Today, I have the pleasure of introducing Dr. Rodrigo Arzate-Mejía. I first met Rodrigo almost 8 years ago now in Mexico, where we both studied genomic sciences. In the last couple of years, Rodrigo has worked as a PhD student in the laboratory of Felix Recillas-Targa at the Universidad Nacional Autónoma de Mexico in Mexico City. There, he established work in Drosophila melanogaster to study 3D organization of the genome. And earlier this year, he obtained his PhD. This week, actually, I think tomorrow, he will be moving to Zurich to start his postdoc at the laboratory of Isabel Mansui. So a very busy year. Rodrigo has gotten several fellowships, which most recently allowed him to complete an internship at Emory University. In addition, he's also a very strong advocate of scientific communication with the public. And to this end, he co-founded the Más Ciencia por México, an initiative which is interested in the promotion of science and technology. We're very excited to hear about your work today, Rodrigo, so please go ahead.
Okay, well, thank you very much, Melvin, for the introduction, and to all the organizers for the invitation. It's my great pleasure. So today I'm going to talk to you about one of the stories that I work with during my PhD at Felix Recillas-Targa laboratory. And this story is mostly about boundaries and genome architecture, and why we believe that these boundaries are actually important, not just to organize the genome, but also to control transcription in space and time. So this story takes place in the nuclei, and as I'm sure all the stories about the seminars take place also in this super happy place, where a lot of different things take place as we are speaking. And I always think about the nuclei as a very crowded space, similar to what we can observe in what we used to observe in the downtown of Mexico City, with a lot of communication and interactions taking place, like craziness. And in particular, I'm very interested in the kind of regulatory interactions that can take place between sequences like enhancers and promoters as to control transcription of genes, specifically during development or in response to specific signals. We know that eukaryotic genomes are very rich in regulatory elements. In many cases, the regulatory elements are way more abundant than the genes that they can target or regulate. And this poses a challenge to the cells as to how to control the flow of information between the different regulatory elements that can be just very close to the gene of interest, and to restrict the communication between those regulatory elements at the genes and those communications that should not really take place. We know from the work of many people that regulatory information between regulatory elements and their target genes can take place just by physical proximity. And by physical proximity, which means that regulatory elements like enhancers are just next to the gene of interest, and they can regulate that. But we also know that in many cases, the regulatory elements can be located very far away from the gene that they actually regulate. I frequently think about the flow of regulatory information between regulatory elements for each gene as a small cocktail party, in which you have a popular guy, which can be your gene of interest, and then you have regulatory elements or people that it's just interested in the kind of information that is flowing in the conversation that is taking place. Of course, you can have people in the cocktail party that are not interested at all in the conversation that is taking place, but you will have other people that it's definitely interested. And a way to get that information is actually to be in close proximity. But by being in close proximity, you can also get to share information with elements or people or regulatory elements that you don't really want to share, because particularly in the genome, that can be a problem. And now imagine that all of these conversations are taking place in each nuclei of each of your cells, not just during development, but as we are speaking and as we are learning. So the cells have developed different mechanisms as to control the flow of information between regulatory elements and the target genes. And one way to regulate that flow of information is by restricting the physical interactions that, for example, enhancers can have with the promoters of the genes. And one way to restrict that flow of information is by the activity of the so-called insulator or chromatin boundary sequences. That essentially what they can do is by interacting each of them in the space, they can restrict communication with the regulatory elements that should be contacting, and by the same time, they can restrict the unspecific regulatory information. During the past decades, we have gained a lot of knowledge about how those regulatory sequences interact in space and how the genome is organized in the nuclear space. We know by this moment that different organisms, all of them shows some extent of three-dimensional organization of their genome inside nuclei, as for eukaryotes, for example. And in all of these cases, like from yeast to mammals to fish, we know that there are regions of the genome that tend to frequently interact in the 3D nuclear space, and that there are sequences that you can observe here in the Hi-C maps that somehow separate these regions of interaction that the people have called them in different ways, like TATs, domains of interaction, contact domains, chromatin loops, etc. But the important thing in here is that we are particularly interested on how does the cell come up with this particular organization? How is that these contact domains are formed? And in particular, this is important because it has been postulated, and we have very nice evidence in mammals, that topological domains can confine genes and their regulatory elements. As you can see, for example, in this picture of typical day in Mexico City subway. And then in this way, the existence of the topological domains can restrict the communication or can reduce the probability of communication with the outside and enhance the probability of communication with the elements within. In particular, in my PhD, we were interested on understanding how does a three-dimensional organization in Drosophila it's formed. And up to this point, there has been different hypotheses as to which could be the main drivers for genome organization in the fly. People have postulated that histone modifications are involved probably through phase separation mechanisms, that the RNA pool itself is involved in this project, that transcription per se is important for bringing together different genomic sequences, and others have proposed that genetic sequences that are bound by architectural proteins like CTCF and many others in the fly are actually important to create these regions of physical interaction. In my project in particular, we reasoned that there are like 4 ways in which you can address which of these different mechanisms are important for the control of gene expression in Drosophila. You can inhibit transcription, DNA, you can deplete DNA polymerase, you can deplete architectural proteins, but in those cases, we can have confounding effects that cannot give us direct evidence of which of them are actually involved in the organization of the genome. So we decided to take a more direct approach and to delete boundaries. But when we decided to delete boundaries, we needed to be careful and as of which gene you're going to use as to perform these experiments, so you can have interesting and clean readout of the possible effects that you can have by deleting these boundaries sequences. So in this project, we asked 2 questions: whether boundaries like these genetic sequences were important to create these contact domains, or they were just a reflection of the histone marks that were located along the genomic sequence, and if they are important for the 3D organization, whether they can impact transcription per se. So we decided to focus on one particular gene, which is a super popular gene, and this is the Notch gene. Notch in Drosophila is located on the X chromosome. It's kind of a small transcriptional unit, no more than 40 kilobases, and it's located on the full transcriptional unit is located in a band of a polyethylene chromosome based on a lot of genetic evidence. We know that the 5' intergenic region of Notch is actually an interband that is visible on polyethylene chromosomes, and that Notch is flanked also by two genes, which are called KIRE and DNC, which are also organized themselves in polyethylene bands. We know that Notch codes for a transmembrane protein, and it's in fact a transcription factor activated by a ligand. And we know, based from a lot of experiments, that the loss of Notch, but the depletion early in development, is lethal, and it's frequently resulting in an hyperproliferation of neuronal progenitor cells. So we know that it's super important and it's quite conserved from worm to humans. But what was most interested for us as to use it as a genomic sequence to study 3D genome organization was one particular evidence that was published probably 30 years ago, or a little bit more, that report a mutant for Notch that had a phenotype. In this case, it was an hypomorphic mutant allelic to Notch that affected eye development. But contrary to most of the mutants have been reported to Notch that affect the coding sequence or its introns, this mutant in particular was located in the interband that was just upstream of Notch and that mapped to the 5' intergenic region of this gene. It was not located in the promoter, and the deletion of this sequence, which was like about 800 base pairs, was sufficient to result in the fusion of the adjacent bands. So back in that time, it was proposed that this could be a chromatin insulator based just on the cytogenic histological evidence. What was very interesting to us was to find out that that the people could revert the phenotype just by inducing inversions or deletion of sequences upstream of Notch or downstream of Notch, which suggested back in that time that probably this interference with Notch function was due to unspecific regulatory interactions with sequences nearby. The final piece of evidence that actually made us decide for this gene was the finding that there is a very good correlation between the interbands or bands of the polyethylene chromosomes in the fly and the organization of the genome into TATs. So we know that interbands are frequently TAT boundaries or domain boundaries, and that the bands themselves are TAT. So we thought that probably based on all what we knew about Notch, Notch itself was a TAT, and that it was that the intraband regions were boundaries. So in order to analyze that, we decided to perform high-C experiments at high resolution in a particular cell line, which is the S2R plus cell line, which is of embryonic origin. And we observed something very interesting. It was very exciting to see that the band patterning that has been reported for this region actually correlated quite well with the presence of domains of interactions or topological domains in the fly. You can observe here that Notch is a TAT itself at 5 kb resolution, and it's flanked by these two huge TAT upstream and one TAT downstream. When we look at 1 kb resolution, we have another very nice, interesting surprise. And it was that Notch was not itself one topological domain, but it was two topological domains, one that was like 30 kilobases and a small one that was about 10 kilobases. And they were separated by two prominent boundaries that we detected by using different algorithms. So this is what you can observe as a cartoon of the organization of this locus in this particular cell type. Then we decided to analyze public data. I'm just going to show you an example that provide evidence that this bipartite organization of Notch is actually present in vivo in data obtained from embryos, and that the organization of Notch is acquired progressively during development, as you can observe in here. First, the first boundary located at the 5' region is detected, and then the other boundary is detected between the intron 5 and exon 6 of Notch later in embryonic development. So based on this evidence, we decided to look also for the presence of architectural proteins, which are frequently bound to the domain boundaries in Drosophila. And here you can observe the binding of different architectural proteins, as well as chromatin accessibility in these two boundaries, which suggests that they can potentially be regulatory elements. We confirm the binding of some of the architectural proteins like CTCF, that it was enriched at the first boundary, and also the presence of histone marks like H3K27 acetylation, which has been shown to be present at the so-called boundaries in Drosophila. We decided to derive a series of CRISPR mutants, as you can observe right here, one that deleted the very first 100 base pairs just upstream of the transcriptional start site that we call the promoter proximal region, then the intergenic region, which contains the CTCF binding site, and then a deletion that removed completely this sequence. And then we also decided to engineer a deletion that was very similar to the mutant that was isolated like 30 years ago, as to compare, as to relate what it has been found in the fly with the molecular characterization that we were planning to do. We took these mutants, we performed Hi-C for all of them in duplicates, and then you can observe here the aggregate results from these deletions. What you can observe right here are the topological effects that we observe upon the deletion of different segments from the 5' region of Notch. The most important finding that you can observe here is that the full deletion of this region that is just upstream of Notch is sufficient to result in the fusion of the first domain of Notch with the region upstream. But interestingly, the deletion of certain regions of this 5' intergenic region is not sufficient to induce the fusion of the domain of the first domain of Notch with the upstream sequence. You can see in here that the fusion of the domain is particularly obvious of the first domain with right the upstream tat of Notch. It was a very clean fusion, and the new boundary is of course located with regions that are bound by architectural proteins. We observe that the deletion of this first boundary result in the loss of RNA pol 2, H3K27 acetylation, as well as the gain of H3K27 trimethylation, and that the deletion or the manipulation of the first boundary did not affect the other boundary of Notch nor another regulatory elements that were located on exon 6. Then we related our findings with transcription, and we found something very interesting. The manipulation of the boundaries do affect transcription, but just of the exons that were located in the very first domain of Notch. The second domain of Notch seems to be acting as an independent transcriptional unit, because as you can observe here, we really don't observe a major change in transcription, while manipulating the first boundary do result in a transcriptional downregulation of these genes. This data suggests that the loss of transcription or the reduction of transcription for these mutants, for example, was not sufficient to fuse the domains, but it was the complete deletion of the boundary, which resulted in the fusion of the domains, but also in complete abrogation of transcription. So that was our finding for the first boundary. And then we decided to look at the second boundary. And the second boundary was very interesting because it was located on an enhancer that has been characterized before for Notch. But contrary to the first boundary that had a lot of architectural proteins, this one has just like 4 architectural proteins bound there. And previous findings have suggested that these boundaries might really not be strong boundaries. So what the people have seen is that probably by deleting these sequences, we might not observe something spectacular from a topological perspective. In any case, we decided to remove these sequences, and we did observe something actually very cool. The removal of this 2.5 kb region was sufficient to fuse or to increase the interaction between the second domain and the first domain of Notch. It did not affect the region upstream of Notch, and importantly, the removal of this sequence did result in the transcriptional downregulation of all the exons now of the Notch locus. Importantly, we lost the RNA pol II binding that we can detect in this region, but also we observe the loss of RNA pol II binding in other regions of the Notch gene itself. We suggest that this region might be super important for the organization of Notch, but also for the recruitment of RNA pol II. Finally, we decided to get more information about this region, and by performing a virtual 4C, we observed that this boundary of Notch, which is located right here, is also interacting with a gene desert that is located 1 megabase away or downstream of Notch. And this was super interesting. We can observe this huge structure right in here, and we decided to look whether it was deleted or lost in our mutant, and we indeed observed that. So the removal of this 2.5 kilobase region is not just sufficient to fuse the domains of Notch, but also to fully disrupt a 1 megabase domain downstream of Notch. This huge topological effect was also accompanied by a loss of interaction for the promoter regions of many of the genes that were located inside this huge domain. And in many cases, we observed that the transcription of many of those genes that were located within this 1 megabase domain of Notch was downregulated in the mutant, as you can observe in this heat map and in these screenshots for the specific genes that were located in this region. So our work actually provides direct evidence that manipulation of the boundaries, as for example for the 5' region of Notch, was just not sufficient to induce the fusion of Notch specifically to the region upstream. It also provides evidence that it can affect the transcription of this gene, and the deletion of the enhancer inside Notch that was acting also as a boundary not just result in the fusion of the domains within, but also in the major loss of a huge domain downstream, which also affected indirectly the transcription of the genes inside. So with this data, we think that we provide, of course, by analyzing a single locus, evidence that in Drosophila there are genetic sequences that can really affect the three-dimensional organization of the genome and can impact transcription. So we published this paper in NatCom. If I don't have more time to get into some details, I think that we really did a lot of analysis. If you have more interest on our work, you can check it out here. So I just would like to thank to Felix and Mayra. Felix was my main advisor, but Mayra was just fundamental to make this to work. Mayra Furlan is a group leader in UNAM. And also thank you to all of the co-authors. It was at Felix Lab, and that's all. Thank you.
Excellent, Rodrigo. Thank you very much for that fantastic talk. I just want to remind people, if this is your first time joining us, you can raise your hand if you want to ask a question for Rodrigo live, and we can give you, oh, okay, so we already have one quick question. And then we allow you to talk, and then you can ask the question to Rodrigo. Otherwise, you can write down your questions on the Q&A, and then so we can ask it back for Rodrigo. So if anybody wants to participate, just raise your hand. So we already have a couple of questions. While people get ready their own questions, I wanted to just quickly ask Rodrigo, so you showed very beautifully this at the beginning, how people almost 30 years ago saw this difference in the eye development of the fly. So now after your work has been done, I mean, what do you think how that adds to that, and how's the explanation for that phenotype now?
So what I think is happening with Notch, because we don't have a complete loss of transcription for our mutants. It's like a very important reduction in transcription. That phenotype in the eye, based on genetic sequences, suggested that we had an hypomorphic mutant for Notch. And what I think that the explanation for that particular phenotype is that the loss of the boundary is interfering with the normal transcription of Notch, but it's not completely disrupting the transcription. Because if you will have something like that, the fly will die just like super early in development, as we know it happens with the deletion of notch. And I did not show you that, but all the computational analysis that we did, they are super in agreement with the genetic available data of inversions and deletions of the upstream sequences that have shown that, for example, the fusion of the first domain of notch with the upstream TAT, the new boundary, let's say, of this huge domain, when you remove that by genetics analysis, it can reestablish the notch transcription in the fly. So it might be that that eye phenotype, it results from a unspecific regulation of notch itself from the new boundary that you gain in the upstream domain. And we are planning to do some additional in vivo experiments recreating these specific deletions to really observe in the fly what happens when we specifically mess up with this detail the boundaries.
Okay, perfect. So I'm going to run a couple of quick questions that we have here from the public. First of all, Irene Kaplo, she's asking, Rodrigo, that was amazing. How common are genes with multiple boundaries?
How multiple they are. So they are very common because we observed that when we analyze that data, we had like 100 genes that were located, like the complete gene within a contact domain. That means that most of their genes are located within that TATs can have multiple genes, or that a single gene can have multiple boundaries. For example, the KIRE gene, which is located just upstream of notch, and you have like these huge TATs, it actually is a huge gene, and you have a boundary that it's right at the middle. And sometimes that correlates with the presence of alternative promoters. And that has been proposed that also promoters can induce the formation of boundaries. So I will say that they are that you can have this very common in the fly gene.
Okay. All right. So Joaquin de Navasquez, I hope that I pronounced that correct. It's beautiful work, Rodrigo. Has any of this been done outside of the S2 cells? Are you using whole flies? I think that maybe you're not, so in which case my question would be whether some phenotypic effect was detected in the cells. Reduction, for example, reduction of MYC might affect anything related to growth or cell cycle.
That's a very good question. So we did all of this just in the S2R plus cells. We derived specific protocols to make it work like specifically. We do observe that the cells have some proliferation problems that we did not keep on working on that. It's very likely that this deletion of the enhancer that can also affect MYC and other genes might have pleiotropic effects. We are planning to do all of those analyses in the future. So it will be very interesting to see whether the disruption of this boundary have phenotypes that are directly related to notch or that are due to the changes, unspecific changes that affect the genes that are downstream.
Yeah, that makes sense. So one last question, Rodrigo. So real quick, Elena Gomez Diaz, she's saying, superb seminar, thank you. I wonder if Rodrigo knows of any study that instead of manipulating topology, manipulates transcription in order to test whether the gene activity affects TAT organization.
Yes, Victor Corses has done a lot of that, and also Juan Manuel Vaquerizas. They both have data that suggests that transcription can affect the 3D organization of the genome in the fly. Whenever you disrupt transcription, you have a reduction in intra-domain interactions and a reduction in insulation, but you don't observe the fusion of the domains, which is what we observe. So my hypothesis is that both you have the transcription that induce the to some level of physical interactions, and then you have also genetic sequences that can add to that insulation. But the thing is that I think that we don't have yet a working mechanism as for how the fly genome is organized in the nucleus space as people in mammals already have.
Yeah, that makes sense. Well, okay, so I'm gonna stop here right now. If anybody wants to continue talking with Rodrigo, he's going to stay in the coffee after both of the seminars are over, so stay tuned. Thank you, Rodrigo.
Thank you.